The previous workflow, pathway analysis, provides a list of ranked metabolic pathways producing a molecule of interest within a selected chassis organism. The next step is to engineer all or some of these pathways. The basic assembly workflow generates assembly plans for plasmids using the basic assembly technique, which stands for Biopart Assembly Standard for Idempotent Cloning. This workflow takes as input a pathway and generates Python scripts to operate an open-trans liquid handler robot, which will perform assembly and chassis transformation. This workflow hence provides a direct link between machine-enabled design and automated assembly. Briefly, the workflow execution is as follows. From the reactions described in the SBML pathway, the cell enzyme node searches for enzymes within the UniProt database for each reaction. The basic design tool converts the SBML file into a set of CSV files listing the DNA parts to be included and enumerate possible combinations of promoters, RBSs, and enzymes into constructs. Finally, the DNABot node generates the set of Python scripts to build the plasmids using an open-trans liquid handling robot. After downloading these scripts onto a computer connected to an open-trans, the user can perform the automated construction of the plasmids at the bench. To run the genetic design basic workflow, go to Shared Data menu and select Workflows. Then choose Genetic Design Basic, click on it and choose Run. Okay, now we can set inputs for the workflow. The first input is the metabolic pathway one wishes to design. The retrosynthesis workflow produces several pathways, but the pathway analysis workflow scored them. Then, it's easy to get the pathway with the best score. The second parameter is the taxonomy ID of the host where one wishes to produce the target from. 83333 is the strain K12 of E. coli. One can also filter the enzymes found to catalyze the reactions of the pathway by indicating one or several particular organisms. We can now set all parameters of each step by clicking on different node of the workflow to modify them. Okay, so we can click now on Run Workflow and see its execution in the history on the right panel. At the end of the execution, one gets Python scripts that can get directly used to pilot the open-trans robot. The scripts can be downloaded and viewed in a code editor.